Welcome to springlib.
To answer this question, we first have to distinguish between row-dependent termination and row-independent termination. So the main difference between row-dependent and row-independent termination, and of course this is in prokaryotes, let's remember, this is in prokaryotes. The main difference is that with row-dependent termination, we are going to use the association between row. This is a factor. It's not necessarily a protein itself, but it's just a factor, a specific factor. You can also consider it to be a transcription factor. It's a specific transcription factor called rho, Greek letter rho, and it is going to bind to the rho binding protein. So it forms a complex with the rho binding protein. And in forming this complex, it will travel up our strand of DNA that is being transcribed into RNA by RNA polymerase, and it will cause, so it causes our RNA polymerase and a lot of the transcription machinery bound where the RNA polymerase is transcribing at that site, at the transcription site, a lot, not only the RNA polymerase, but the other transcription factors that are bound. So we can call this the transcription machinery. It will cause the row-dependent termination. We will see that the rho factor will cause the RNA polymerase, and like we said, all these different transcription factors and transcription machinery to dissociate. So it will dissociate from our DNA strand being transcribed. So this is how, in effect, our transcription is going to be terminated with row-dependent termination. Whereas if we look at row-independent termination, once again, in prokaryotes, we don't have rho, but instead this could actually be to a few different factors. We actually can form this hairpin. So the formation of a hairpin, and let's just clarify that the hairpin will form due to the way that the RNA that is transcribed from the DNA, the way that it will emerge from the RNA polymerase, and the way that it will basically go into a tertiary structure, where it will go into interactions with the base pairs and between the base pairs, and they will cause this RNA strand to go into a particular conformation that produces a hairpin. And this hairpin formation causes dissociation of our DNA strand from the RNA polymerase. So the end result is the same. Our transcription machinery, which includes the RNA polymerase and transcription factors, will dissociate from the DNA, but the way in which we get to this point is different. With rho-dependent termination, we are using rho, which is this factor that will, when bound to the rho binding protein, it will travel up this transcription, travel up the DNA that's being transcribed. It will reach the transcription site and cause for the RNA polymerase and other transcription machinery to dissociate. It kind of pushes off this machinery. You could think of it in that way. Whereas with row-independent termination, it's actually due to the RNA strand that is produced itself. So the product of our DNA transcription is going to be the cause for the dissociation of our RNA polymerase from the DNA strand. And just one more point to add here is that the hairpin formation is the DNA that it is going to be the RNA. It is the RNA that emerges from the transcript. So once it's been transcribed. So from the RNA polymerase. So it is, once again, the RNA that emerges from the RNA polymerase once the DNA has been transcribed, and it will go into this hairpin formation, which at a certain point will cause our RNA polymerase and other transcription machinery to dissociate from the DNA strand. So, looking at our question, going back to our answer choices, we find that the correct answer is going to be choice A. So our answer is choice A.